Good afternoon, everyone. So we give it a few more minutes. So I think we're ready to start now. So I wanted to thank you all for being here, either virtually or in person. So to attending this technical seminar from 10X Genomics. Today we'll be discussing the fixed RNA profiling assay, which we have available for human and just newly on mouse as well. So really looking forward to be able to present to you the advantages of this assay. And thank you again all for being here. I'd like to introduce Egon Ranghini, which is our science technology advisor at 10X Genomics and has been at the company for 5 years. So thank you, Egon.
Thanks, Marie. Hello, everyone. Thanks for joining us today. Thanks for the invite. My name is Egon. I'm the science and technology advisor for 10X. I apologize if I look at the camera and don't look at you guys, or vice versa. I can try to alternate. Yeah, sorry about that. Couple of things before we start is I'll be more than happy to share this presentation with you at the end of the talk. And if you have any questions, just feel free to raise your hand, interrupt me. We can more than happy to take questions at any time. So for today, I'd like to talk about our newest solution, that is the fixed RNA profiling kit. Now, before we go into that, I just wanted to quickly show you what we have at 10X. That actually, let me go back here. We've been developing those 3 platforms to unpuzzle the complexity of biology. And the Chromium platform is what we have for single-cell genomics, and what we're gonna talk about today. And then if you guys have a question about our spatial transcriptomics platform, feel free again to ask. We can also address that in case. And so what I like to say is that we are very proud to see that this technology has been used in over 3,700 publications now, 3,700 studies, and this number keeps growing. And at the same time, we are also very excited to witness firsthand how researchers, how investigators have been used this technology to advance not only their research field, but the understanding of biology in general. And when we look at the 10X workflow, we always look at what we call the beginning to end workflow. That's because all reagents that you need to make a sequencing ready library, those reagents are provided in the kit, are already part of the kit we provide. And so when you start a single cell experiment or a single nuclei experiment, all you need to prepare is a high quality single cell or single nuclei suspension. And then we also take care of the bioinformatics side, where we provide bioinformatics pipeline for data analysis and data visualization. Those comes at no cost for you. Now, the technology, probably you are maybe already familiar with that, but in case, what we do is to partition individual cells or individual nuclei and individually barcoded gel beads into those little droplets that we call gel beads in emulsion or gems. And in those droplets is where cells are, let's say, lysed, and their content, their mRNA, is released and captured by the gel beads, by oligos that are attached to the gel beads. And so when we first launched this, the single cell gene expression solution back in 2016, we were only able to measure gene expression, and so capture RNA and look at our transcriptome. That was great, but we know that biology is very complex, and so over the years, we enriched this offering, and so now we are able to capture multiple types of information simultaneously from the same cell. This is essentially what we call multi-omic cytometry, and it's really a way to better understand the complexity of biology. That's again because the more information you capture, the more data you have, and then you can get a better understanding of the biology of the sample you are studying. And so, for instance, we can now co-measure gene expression, the expression level of cell surface protein. We can look at CRISPR-mediated gene perturbation. We can profile the full-length VDJ sequences of T cell, B cell receptor. We have a solution that allows us to look at gene expression and chromatin accessibility from the same cell. And so, really, depending on what you are studying, what questions you have, we pretty much have a solution for to answer those questions. And there is a paper that I really like presenting pretty much every time I give a talk, which is this one, that really shows the power of combining multiple type of data set to answer a complex question. So in this case, in this paper, the authors wanted to kind of better understand why COVID symptoms oftentimes seems to be milder in children compared to. Sorry. We are not in the Zoom meeting. Interesting. Okay. We are in. Oh, okay. Okay. So, yeah. So I was saying, so the goal here was to essentially understand why COVID symptoms sometimes seems to be milder in kids compared to adults. And so, and to address this question, the authors profiled 700,000 cells. That's a very large study. They were isolated from pediatric as well as adult COVID patients. And they generated a true multi-omic data set in the sense that they generated gene expression libraries, then those TCR and BCR or VDJ libraries, and they looked at expression level of cell surface protein. This information came from the same cell. All these different pieces of information came from the same cell. And thanks to this wealth of information, the authors were able to demonstrate that the adaptive immune system of children showed a significantly higher clonotype diversity. Now, it might not be very surprising, but this result was made possible. They were able to essentially obtain this result by, again, looking at multiple types of information at the same time. Now, we know that, I mean, this is great again, but at the same time, we know that working with living cells or living tissue can be challenging. And that's because we can encounter situations in which samples might not be available during normal business hours, or perhaps we want to collect samples over multiple time points before profiling them. Of course, sample degradation can be an issue, and this is sample dependent. There are some cells, some tissue that really don't like being dissociated and don't like sitting on ice. And so what we really want to prevent here is sample degradation and maintain high viability. So I guess this point here, sample stabilization is crucial. That is probably one of the most important part of the entire workflow. And then, of course, we can encounter some logistical constraints where perhaps you want to ship samples to a collaborator or receive samples from a collaborator. And so to essentially address all these issues, very recently, 3 months ago, we released this fixed RNA profiling solution, which, as the name suggests, allows you to collect your samples, fix your samples in 4% formaldehyde, or PFA, I should say, and then store your single cell suspension or your single nucleus suspension at -80 for up to 3 months. Now it's actually up to 6 months. We just extended, which essentially allows you to block the biological state of your sample, preserve the biological state of your sample, really, no matter when you collect those samples or where you collect those samples from. For this kit, we use a probe-based chemistry in which we target approximately 18,000 coding genes in human and in mouse. And here we can see how what the chemistry looks like essentially. So we have a set of probe pairs. They've been designed to account or to detect all annotated isoform of a given gene. And we have a left-hand side and right-hand side probe. So attached to the left-hand side is a partial read-to sequence for the Illumina flow cell, and attached to the right-hand side probe is the capture sequence for the gel beads. Now, the target sites that you see here, this is 50 nucleotides. And so even when the mRNA is degraded because of the fixation process, we can still detect those transcripts, we can still detect the gene expression. And so what happened here is we have approximately 55,000 of those pairs. They will target more than 18,000 coding genes in human and mouse. As you start the workflow, those two probes will align to their target size, then they will ligate. This is absolutely essential in order for the probes to be detected. And it's a step that essentially increases the specificity and sensitivity also of the assay. And so because of the probe-based assay, we now have a we can actually see a significantly higher sensitivity when we use the fixed profile compared to the traditional 3' chemistry for, let's call them fresh sample or sample that have not been fixed. And those results have not been cherry picked. So that is pretty much what we see across all samples that we profile. Now, this is internal data. There is also some external data, and there is this great bioRxiv manuscript. It's from Luciano Martellotto and Jasmine Plummer. And what they did here was to compare, again, the fixed chemistry against the 3' chemistry on the same sample. And they found that the fixed chemistry could detect more than twice the number of transcripts compared to the 3' chemistry. And importantly, they also showed that the fixed chemistry could increase the detection of lowly expressed genes, including transcription factors. Here we see the top 20 expressed transcription factors. So it's really a chemistry that allows you to improve the sensitivity or get a better sensitivity compared to 3' and also the 5'. One thing I didn't mention, and apologies for going back, because of the probe-based chemistry, we can also reduce the minimum recommended sequencing depth to just 10,000 read pairs per cell. And this is as opposed to the 20,000 read pairs per cell that we recommend with 3' or 5'. So you're going to also be able to save some money when sequencing these libraries. Now, I'd like to show you some slides regarding, consideration when using this chemistry. So first of all, because it's a probe-based chemistry, we have probes for human and mouse. So if you use a different model organism, well, we can have a conversation. It's possible, it's doable. It's a kind of one of those off protocols applications. But that approach will entail designing custom probes and spike them in. Again, it's not something we have a protocol for, but it's doable and it's been done. And now in terms of sample compatibility, this is essentially what we can fix. So we can fix single cells, we can fix single nuclei, we can fix tissue, which really increases the flexibility of this kit because now you don't need to extract cells anymore. You can just fix the tissue and then worry about isolating cells later on. And we have also a protocol that allows us to profile single cells from FFPE blocks. And I'll show you some slide later. Now also prepares this slide that is intended to be kind of maybe not a decision tree, but it shows you what you can do depending on what samples you have. So first of all, we have demonstrated protocols for fixation. So you don't need to really optimize or find a protocol online, just use our protocol. If you start from single cell or single nuclei, you will be fixing those cells or nuclei, and then you can store them for up to 3 months. Well, actually now it's up to 6 months. What if you have a tissue? So fresh tissue. Here is where you can dissociate the tissue into a single cell suspension and fix those cells, or you can isolate nuclei from tissue, and we have a nuclei isolation kit I'm going to talk about later. Or even better, I would say, you can mince this tissue and then fix the minced tissue, store it for up to 1 week at 4 degrees, and then isolate cells. And once you isolate those cells, because cells have been fixed, you can store those cells for up to 3 months. And similarly, you can do that with the frozen tissue. You can decide to isolate nuclei using the nuclei isolation kit and fix those nuclei, or you can mince this frozen tissue, fix the tissue, again, store it for up to a week at 4 degrees, and then isolate cells. So it's really you have a lot of flexibility, and I'm finding more and more, it's difficult for me to find a tissue or a situation or tissue or something or a sample that cannot be fixed. I'm sure there's something that we cannot do, but so far it's pretty much we can fix almost anything. I don't want to say anything, but almost anything. Now, there are also some other considerations we need to keep in mind. So because of the washing steps, when we start with cells, we want to start with at least 300,000 cells for the fixation process, 500,000 nuclei if you use nuclei. If you want to fix tissue, it's a good idea to start with at least 25 milligrams. The more, the better, but 25 milligrams is what we've found in-house that works fairly well. Now, when it comes to sample dissociation, of course, when you have fresh tissue, you want to ensure that if you isolate cells, those cells stay alive. You don't want to fix dead cells. But if you fix the tissue, that concern is no longer, I mean, that is no longer a concern because we fix the tissue, so viability is no longer a problem. Now, I mentioned that samples would need to be fixed in 4% PFA, that is the final concentration of the fixative we use. There is also a little kit that you need to buy from us, which is a sample prep kit, and it contains a couple of items that would need to be used during the fixation procedure. So that's to say that, you know, it's we can't really simply take cells and fix them in 4% PFA. There is also this kit that will need to be purchased from us. And one last important consideration here is what do we do when we count cells or nuclei that have been fixed? So it's strongly recommended to use fluorescent instead of trypan blue when you count your cells or nuclei after post fixation. That's because if you use trypan blue, for instance, if there is debris, trypan blue will stain also debris. Whereas when you use fluorescent, that is the count will be way more accurate. So strongly recommended to use, for instance, Ethidium homodimer 1 or Propidium iodide. And this is part of the protocol. Now I'm going to show you some data we generated in-house on this chemistry, using this chemistry. So first of all, this is human PBMCs. What we did here was to fix PBMCs for different length of time, up to 6 months, and those samples were stored at -80. And you can see that there is no really much difference between, actually there is no difference between the different conditions we tested. So cell frequency, cell population are not impacted by the fixation procedure. Here we have a human colon sample. In this case, we fix the tissue. So we collected a fresh tissue. This was chopped, minced, fixed, and then we immediately isolated the cells. And those cells were profiled, and we were able to identify the main population of the colon, including specific cells such as goblet cells here, myofibroblast, neurocrest, enteric neurocrest cells, and also these mast cells. And here we did something slightly different. So we obtained a human endometrium sample. That was a fresh sample and then a snap frozen sample. We again fixed the fresh or the snap frozen, what we chopped, minced, then fixed. And then the 0 day here means that the tissue was immediately processed for single cell isolation and profiling. 5 days means that the tissue was stored at 4 degrees for 5 days before we isolated single cell and profiled those cells. And as you can see, again, there are very little differences. So bottom line is all the different conditions really don't affect the type of cell or the cell frequency that we are able to recover. And the fixed RNA profiling is also a multi-omics solution in the sense that you can use oligoconjugated antibodies to detect the expression level of cell surface protein, and then of course gene expression from the same cell. Just keep in mind that if you follow this protocol or this strategy, you will need to label cells before fixing them, and the sample will need to be run in a single plex format. So we won't be able to multiplex them together. I'll mention multiplexing in a minute, but just keep in mind those two things. And then we also have preliminary data showing that we will be able to detect intracellular proteins. So this is great because so far, if you look at our 3' or 5' solutions, we have methods to detect expression level of cell surface protein, but not intracellular protein. Now with this fix, we will also be able to do that. The protocol is not available yet, but it will be available soon. Now, sample multiplexing. This is the other great advantage of this kit. So you probably are familiar with the concept of cell hashing, where essentially you label different samples with antibodies, for instance, and then you combine those samples together and run them in the same channel of the microfluidic chip. That is a strategy to, let's say, reduce the cost of your 10X experiment, because at that point you'll be running only one gel bead reaction instead of multiple. And also you'll be able to detect a certain percentage of multiplets. Now, with the fixed RNA profiling kit, we can multiplex samples without using any labeling. So no antibodies or no oligo-conjugated lipid molecules. And that's because we included a unique barcode to each set of probes. And by using the information from the barcode during data analysis, we can demultiplex samples. So here the advantages are, well, first, you don't need to buy antibodies anymore. Second, you don't need to decide upfront which sample you want to combine together. Here, simply, you collect your samples, you can store them in -80 for months. And then when you start your experiment, you combine the sample the way that makes most sense for your experiment. Not when you collect the samples, but when you start the single cell workflow. So you have essentially 3, 4, 5 months to think about whether or not you want to combine the samples. So with the largest size of the kit, we can combine up to 16 samples together in the same channel. And this will allow us to target 128,000 cells per channel. A microfluidic chip has 8 channels. And so if you were to run a full chip, that will allow you to target 1,024,000 cells per run. So it's really a large study that you can potentially do. And of course, when you combine, I'll show you a slide later, but when you combine samples, that is also where the cost saving comes into play. A significant cost saving comes into play. So here, what we did was to collect splenocyte from a mouse. There were 112,000 cells in total. We split this sample across 16 pools and then combine them in the same channel. And as you can see here, those UMAP projections are essentially identical, which shows lack of batch effect due to the multiplexing. Then we also ran this experiment in-house, which I'm hoping is going to show you the sensitivity and the flexibility of this kit. So what we did was to essentially follow a very well-characterized assay in which we looked at activated T cells. And this happens when you incubate resting T cells with beads conjugated with anti-CD828 and anti-CD3 antibodies. And so resting T cells becomes activated T cells. For this experiment, we used PBMCs from 2 independent donors. We stimulated these cells for up to 2 hours. So it's 5 minutes, 15 minutes, and then up to 2 hours. We included 2 controls. And so in total, we had 16 samples, which is a great number, the perfect number to combine those samples together. We pull them together and run them in only one channel of the microfluidic chip. And then we looked at the data. And so first, we look at the expression of FOS, which is a marker for T cell activation. There is no expression in the T cell in the control at the beginning or at the end of the experiment as expected. But then if we look at the stimulated cells, we start to see the expression of FOS already at the 5 minutes time point. And I believe the expression peaks around 60 minutes or so. CD69 is another early marker of T cell activation. Again, the expression was very low in T cell, and then as expected, the expression peaked around 60 to 90 minutes. And then we looked at VISTA, which is a immune checkpoint marker, low expression in the let's call resting T cells or unstimulated T cells. And then as expected, again, the expression started to increase around perhaps 15 minutes, I would say, and then it will peak around 60 to 90 minutes. Now, I'm not showing you any groundbreaking biology here. This is really a well-characterized model, but I'm hoping that this data shows you how flexible this kit can be, or the flexibility that enables. So now you can collect sample at multiple time points and don't worry about keeping those samples alive for hours. You can immediately fix them. Viability is much less of an issue in this case, and also the sensitivity because now we can immediately detect the expression already after 5 minutes. Now, yes, sure.
So how long
Oh, so good question. So I'm gonna.
Thinking of this, right, you've got samples coming off of the fire cat
Yeah.
and so on. So.
Yeah, we did.
If you guys stuck them on ice and then put them all together?
No, so it was, so the question is how long the fixation takes. So we fixed each sample. I believe this was the overnight fixation of 4 degrees, and then we didn't run the experiment at the same day. That was run perhaps the following day or the day after that. But each sample was collected, was fixed. Or the fixation protocol started in this. And when it comes to the fixation protocol, you have two options. You can fix the samples 4 degrees overnight. That is the best solution, the best approach, I would say. Or you can fix the sample for 1 hour at room temperature. The important thing is if you start, let's say, with a protocol, let's say 4 degree overnight, and then you collect multiple samples, just stick with the same fixation procedure.
So here we just added the paraformaldehyde.
Yeah, we just say, yeah, we had,
And stuck it in the.
yeah, and then either the next day or the next week that cells were profiled. Okay, now we also have this really exciting application that we can do with this chemistry, and we have now the possibility of isolating single cell from FFP blocks. So this is a protocol. It's not a different kit. It's a cell dissociation protocol. Actually, we're going to have two protocols. Now we have only one. But in a nutshell, what you will be doing is to prepare a couple of FFP curls out of your FFP blocks, and then either use either a manual dissociation method or what is available now, a modified protocol which uses the Miltenyi dissociator to isolate single cells, and then you can profile single cell with the fixed RNA profiling kit. Now, we tested various tissue types in-house. I believe we have more than 20 now, and the oldest sample tested that gave good data was a 14-year-old human lymph node sample. The protocol we have available now, as I mentioned, is a modified version of the Miltenyi protocol that uses the Octo dissociator. And you can download it from our website, or if you scan the QR code either now or when you get the slide, just let us know and I'll send you the protocol. The manual dissociation protocol, which will not require the GentleMax dissociator, will be available soon. It's not available now, but it will be available soon. And so we generated some internal data. This is a lung cancer sample. It's a human lung cancer sample. We prepared two 50 micrometer scrolls and use the Miltenyi protocols to isolate cells. And as you can see here, we can detect multiple classes of type of cells, including, here we have plasma cells, NK cells, we have lung-specific cells, such as those alveolar cells, and also the cancer cells. But what I think is more exciting is to see that this technology has already been used successfully in the field. This is the same manuscript that I showed you before, so it's still from Luciano Martellotto and Jasmine Plummer. And in this manuscript, in the second part of the manuscript, those authors developed a method to isolate intact nuclei from FFP blocks, and then use the fixed RNA profiling chemistry to profile those nuclei instead of profiling individual cells. And I love the quote from Luciano here, or his tweet, where he said that the cure for cancer is sitting in a pathologist drawer. And so what they did here was to essentially develop this protocol that allows them to isolate intact nuclei from FFP blocks. And then they profile those nuclei with the fixed RNA profiling chemistry and compare the data with a matched sample that was profiled using the Visium for FFP solution. And Visium is our spatial transcriptomics platform. And they found a strong correlation in terms of genes detected and also in terms of the cell types detected between the two technologies. And again, the last bullet point here, I'm paraphrasing again, I'm paraphrasing now at this point, Luciano, he says that this technology now enables high quality and sensitivity single nuclear RNA sequencing data from FFP tissue, and essentially now allows investigators to generate extensive or perform extensive retrospective genomic studies using their archived FFP sample. So this is extremely exciting because for the very first time, we have the opportunity to generate single cell data and spatial transcriptomics data from the very same tissue block, which will give us an unprecedented view of the biology of our sample. And again, super exciting, I believe. Okay, do you guys have any questions? Yes.
Are there following metrics?
That's a good question. So the quality metrics are the same. I would need to double check, but what we see in house when we isolate the FFP cells from FFP blocks is that there are a good number of tissue that really generate high quality data. Sometimes that quality is not as high as fixing a tissue directly, but it's really sample dependent. And so if it's not neck and neck, might be a little lower, but you're still able to generate high quality data. And
So are we still dealing with sort of a 200 and higher than
so
30,
that
or?
yeah, so that's a great question. And so the V200, the answer is not yet, because our R&D team has started looking at the V200 correlation also for FFP, but they need to look. So they haven't looked at enough tissue as of yet, and so they basically say at this point we don't really have guidance when it comes to the V200. Of course, there is a higher chance that if you get a high V200, you could probably get a better data. But we don't have now a direct correlation because we need to look at essentially more tissues in order to find a more strict guidelines. So for now, my recommendation would be if you have if you can actually spare some section, I would probably do the V200. And if you are above 50%, that would be probably fine. If you are between 30 and 50%, you might be fine. Depends on from tissue to tissue. If you are in the low teens, that might be more challenging. So yeah. But I'm also hoping we're gonna have some more specific guidelines soon. Okay. Oh, and then one important thing is so we generate the data. If you want to analyze the data using, let's say, using our cloud, this is a possibility to essentially run our cell ranger pipeline for free. And you don't need any bioinformatics experience or access to a Linux machine. Just create an account with us for free. You can upload the data, FASTQ data, and it features a GUI. So it's really button and drop-down menus that allows you to run very quickly the bioinformatics pipeline. Oh, and then yes, this one. So this is a slide that I wanted to show you to essentially show how you can save money when you run the fixed RNA profiling compared to the 3' chemistry. Now, this is only 10X reagents, so it does not account for core fees or for sequencing. It's just 10X reagents. 3' reaction, 3' kit is about $1,600 per sample. When it comes to the fixed kit, there are 3 sites of the kit. The smallest is a 4 reaction. Essentially, you can run 4 samples and you cannot multiplex them. It's a little bit more expensive per reaction, but think about the flexibility and think about the increased sensitivity. Now, when you start multiplexing samples together, that is where you start saving money. So the mid size of the kit is a 16 reaction. You can pull together up to 4 samples, and so for a total of 16 reaction. And then the cost per reaction is about $1,300. So you already start to save $300 per reaction. And then if you use the largest size of the kit, which is this, which allows you to put together 16 samples, for a total of 64 reactions in total for the kit. That is where, if you take full advantage of the kit, each sample will cost you only $550. Again, 10x cost only, not core, not sequencing. But at that point, and I like to quote one of the core directors we've been working with, at that point, the cost per sample is pretty much what you were going to pay a couple of years ago for bulk RNA sequencing. So it's really making this technology very accessible. And again, think about flexibility, think about increased sensitivity. Now, switching gear, the other kit, the other new solution that we released recently is the nuclei isolation kit. And there are some reasons why we want to isolate nuclei. So for instance, we might have a frozen tissue. Now we know that we can also fix the tissue, but frozen tissue where we need to isolate nuclei, there are some cell types that are better preserved, better represented if we isolate nuclei. So think about adipocyte or human podocytes, kidney cells. And then of course, there are assays that do require isolating nuclei. So if you are interested in chromatin accessibility, if you want to look at the region of accessible chromatins, the input material would be nuclei and it would not be whole cells. Now, there are also challenges with isolating nuclei, and that is because there are many protocols available online, but those protocols are oftentimes cell-specific, tissue-specific, condition-specific. You will need to optimize the analysis conditions to make sure that you have good quality nuclei. And typically, many of those protocols will also require cleanup steps where, you know, either you use a fax or you, or in any case, you might end up with less material than you start with. And believe it or not, but oftentimes when it comes to optimizing nuclei isolation from tissue, that process might take anything between a couple of weeks or a few months. So we wanted to come up with a better solution. And so we released a nuclei isolation kit, which is compatible with a large variety of tissue, frozen tissue, and also work with fresh tissue. All it takes is 3 to 50 milligrams of tissue and 1 hour of your time, and requires only a benchtop centrifuge. And in a very 10x fashion, all reagents that are required for the nuclei isolation, including the RNase inhibitor, are already part of the kit. And so here we this is to show you how the kit has been optimized in order to remove most of the debris and the aggregates. So here we isolated nuclei from an adult mouse brain. If you were to look at the sample immediately after a dissociation, this is how it will look like. So it's essentially full of debris and full of clumps. If you filter it, even with a 70 micrometer, you still end up with some debris and clumps, and this will definitely affect the quality of the data. But if you go through the entire workflow, that is where you will be generating a high quality nuclei suspension. Now, if you work with brain, you might still see some debris, some small debris. That is acceptable, but you're more than welcome to send us a picture so we can take a look and make sure that everything looks fine before you proceed. Here, for instance, we generated multi-omic data, so gene expression plus ATAC data from the same nucleus, starting from a human kidney tumor sample. And we were able to identify not only the main population, cell population present in the kidney, but in particular also those podocytes. And I'm a kidney guy, so it's very exciting to see that we were able to capture so many podocytes efficiently. And that's because podocytes in human are huge, are very large, they have a lot of processes. So sometimes it might be tricky to capture them if we use whole cells, but when we use nuclei, well, the size is no longer an issue. So we can really capture those cell types very efficiently. And we can fix those nuclei. So we can isolate nuclei with this kit and then fix them and profile those nuclei with the fixed RNA profiling kit. And when we did that and compared again the sensitivity of those using the fixed RNA profiling solution against the 5' chemistry this time, again, we found a significantly higher sensitivity with the fixed RNA profiling kit as compared to the fresh or 5'. And also the data quality was slightly better, this is represented by the fractional reads in cells, which indicates the level of background that you have in your assay. So it's mid to high 80s or 80 to 90%, I should say, versus mid to high 90s. So better single cell data essentially. And a question that we often hear in the field is, okay, I isolate nuclei, I profile nuclei, I no longer profile cells. What type of information am I losing? Because the entire cytoplasmic is gone. And the answer is, yes, you'll be detecting less genes per cells and less transcript per cells, because again, the entire cytoplasmic compartment is gone, but the biological information is retained. And so this is, we have been seeing this across multiple papers where essentially running cells or running nuclei, you will be still able to detect the same cell population with pretty much the same frequency. This is PBMC data, this was PBMC data, but there are also kidney data as well. And also, again, think about cells that might be very large in size. At this point, you can capture them more efficiently. And we also see situation in which heat shock genes are not expressed in the nuclei data. That's because you don't dissociate a tissue with a warm dissociation, for instance. So the biology sometimes can actually be better represented because you don't treat your samples with at 37 degrees, for instance, or you don't digest your sample for an hour at 37 degrees. Now, there are really many solutions now available, and the question is, well, which solution should I use for my experiment? Short answer is reach out to us, reach out to Pat, reach out to Fernanda. We are here to help you. My personal feeling is, I've been starting recommending using a fixed RNA profiling solution as the default, unless you, well, of course you need to have human or mouse, okay? Unless you want to look at chromatin accessibility, in that case, we'll need to use the multi-omic kit, or unless you're interested in profiling VDJ sequences, or unless you are interested in performing a CRISPR screening, but for a typical gene expression experiment or whole transcriptome experiment, especially if you are using human or mouse, I will definitely go with the fixed RNA profiling solution. It gives you more flexibility and more sensitivity. It makes life easier. And if you combine sample, you can also save money. But again, bottom line is just reach out to us at any point. We are here to make sure you're going to be successful. That is our goal. And one thing I always mention is with 10X, you never get only one call or one meeting or one presentation. So really, we are here to help you, and that's our goal. And with that, I just want to introduce your field team. Eve Marie, she is your sales executive. Let's say the quarterback of the team. Emma, she is your sales associate. She used to work here. The same analogy, football analogy, maybe wide receiver, running back, Robin and Batman. That's even better. I'm Egon. I'm your science and technology advisor. I don't know where I fit in that analogy. Bruce Wayne. Joking. But yeah, that's your field team. And yeah, thanks a lot for your time, for your attention, and I'll be happy to take any question if you guys have any. Yes, please.
So going back to the graph where you were comparing the number of genes when you were comparing the fixed RNA profiling kit to the 3' kits, when you're saying median genes per cell, are you referring to the total the number of genes transcripts, or are you referring to like
Yeah, I think it does. It's the number of genes per cell that we detected.
So in total, and not necessarily like.
No, it's not the total number of genes that was detected in the assay. Is the number of genes that was detected in per cell. So we take the total number of genes, and that it's per cell. Yeah.
So it could be possible that even though the fixed RNA profiling kit is detecting more genes of the cell, it may not actually be more unique. New identified genes that were not previously identified in these 5 times
No, it's yeah, that's a good point. I don't have that graph. So I know what you mean. It's essentially if we, let's say, create a Venn diagram.
Previously able to see with the 3 times.
Yeah, no, that's a good point. So I don't have that data to show you, but we can detect also more lowly expressed genes. So there is a chance, there is a good chance that if a gene is not captured by the 3' or the 5', that genes will be captured with the fixed RNA profile. Yes, but that's a very good point. Any question online?
You want the probe design is splicing, right? So you've got 18,000 genes and 5,000.
Yeah.
Does that count into?
It includes all annotated isoform. Yeah.
All annotated?
Yeah. So yeah.
So there are still some, I mean, depending on where in the estimate of total, there's still some gaps.
Yeah, and yeah, there could be, and so
Mostly like comparative.
yeah. So we don't have probe. For instance, we don't have probes for PCR or for TCR or BCR. We don't have probes for ribosomal proteins because essentially, especially ribosomal proteins, will add nothing to the story, and it will dilute the sequencing reads. But if if you are interested in, let's say, targeting a specific transcript or a specific isoform, that will be one of those, let's say, off protocols application in which we can think about designing probes and spike them in. And it's where also we're already working with some customers who are interested in detecting what either is not human or mouse, and or they are interested in detecting specific sequences, specific splicing variants, or we also hear question about detecting SNPs. That is a little bit trickier, and so again, we can think about spiking in probes, designing probes specific to detect SNPs, but it's a little bit trickier because you might have a mismatch or may have a SNP, but the probe might still bind. So yeah. Any more questions? Okay. Thank you.